Thank you. I was just talking earlier, and last week I was at a public lecture by Alan Alda, the actor, whose big mission today is about communicating science. And one of the first things he said was, get out from behind the podium. So I am out. And now I'm ready to tell you about some of my research, but I've set this in a broader context. I really want to talk about stem cells and genome editing, which you hear a lot about in the public domain and in the literature. And I want to talk about the implications for these developing technologies for precision medicine, so how they can be applied to human health. But I'm going to try to set it actually in the context of some of my own research. And my research over the years has been focused on this embryo here. This is the mouse blastocyst. And this is the stage of development that I first encountered when I was a graduate student with Richard Gardner in Cambridge, along with Martin Johnson. And I fell in love with the blastocyst, and over the years, on and off, I've been working on this stage of development. Sometimes going off and do other things because the tools weren't there, but we keep coming back to this embryo. And the mouse blastocyst at this stage here is the size of a speck of dust. It's got about 100 cells, and it's just about to implant in the uterus. So this embryo then has 3 cell types, an outer cell called the trophectoderm, a group of cells at one end called the inner cell mass, which contains 2 cell types at this point, primitive endoderm on the surface, and this little group of pink cells called the epiblast. And the questions that we wanted to ask were, what are these cells? What do they give rise to? We know that now from many studies, from many people over many years, from the 1970s to the present, with more and more detail. We know that the trophectoderm gives rise to the major parts of the placenta, the primitive endoderm to the yolk sac, and this little group of cells are what we call pluripotent, because they give rise to the entire fetus. So that, then, was what I was working on when I was here in Oxford and Cambridge. And then I left the UK in 1977, married a Canadian, moved to Canada, and started thinking about stem cells. I met my stem cell heroes. This is Till and McCullough. They are the people in Toronto who first identified stem cells in the hematopoietic system. And their concept that there was a self-renewing cell that retained the capacity to make multiple cell types is really fundamental to how we think about all stem cells today. Self-renewal and retaining potential. And then that started me thinking, and others as well, could we make stem cells from the blastocyst? And in particular, of course, people were particularly interested in whether we could make stem cells from the pluripotent cells. And it's lovely in Toronto, outside the Mars facility where the Gairdner Foundation is, we have a statue of Till and McCullough, and here's Jim with his statue. And you can actually sit on there and have a discussion with your heroes. So, what we now know, of course, is that you can get stem cells from the mouse blastocyst, the most famous being embryonic stem cells, which derive from those epiblast cells. Now, we tried to make them. We were not the first to make them. They were derived by Matt Kaufman and Martin Evans in Cambridge and Gail Martin in San Francisco. But they are the cells that behave like these pluripotent epiblast cells. They grow indefinitely in a dish, express specific transcription factors, and you put them back into an embryo, and they contribute to all the cell types of the fetus, but not to the placenta, not to the yolk sac. And my lab made trophoblast stem cells, which continue to divide indefinitely. We made Zen cells, and you can see when you put those back in chimeras, they do what they would have done in the embryo. So we've captured that lineage specification state in these 3 cell lines. They make endoderm, they make placenta, and it's these cells, of course, that in the end have been the most famous. The other ones are great, we do lots of good things with them, but embryonic stem cells, of course, have this capacity, apparently, to make all cell types in a mouse. But were they really pluripotent? If they were, then ES cells alone should be able to make a mouse, because that's what the epiblast in the blastocyst can do. So my colleague Andras Nagy and I did a bit of genetic trickery in what I still think is my most interesting experiment ever, because what we did was we said, well, if ES cells can make the fetus, if we combine them with an embryo that can make trophoblast and endoderm but can't make the epiblast, then maybe we can have a complementation system whereby the ES cells can actually take over the fetal compartment. And that's what you see here. It's the tetraploid embryo, green, ES cells, blue, put them together, entire fetus is ES derived. But more excitingly, of course, is if you let those go through, then you can get whole litters of mice that are perfectly viable. They're all GFP expressing this time around, and they are entirely derived from ES cells, from cells that have been grown for many generations in a petri dish. And to me, that is still a pretty amazing thing to see. But it was those papers where we demonstrated the true pluripotency of mouse ES cells that really underlie then the concept that if you can do this in the mouse, then what if you could do it in the human? Because now you would have a stem cell line in which you can have an endless supply of cells to make every cell type in the body and potentially use it for regenerative medicine. And of course, again, we know the history. In 1998, Jamie Thompson in Wisconsin was the first to derive human embryonic stem cells. Same thing, as we do in the mouse. We take the blastocysts and derive these cell lines. And indeed, in cell culture, human embryonic stem cells can now differentiate into many, many different cell types. But what was more exciting still and really transformational and leading on to this sort of precision medicine connection that I want to talk about was the discovery by Shinya Yamanaka that you could actually do away with embryos and take adult cells, both mouse and human, skin cells, blood cells, other cell types, and add back, express in those cells some of the genes that we and others had shown were important for the pluripotent state in the embryo, and you could turn back the clock and turn adult cells into embryonic stem cells, essentially. These are called induced pluripotent stem cells, and they can differentiate in culture. And now this opens up the possibility of personalized stem cells. And now we're into the beginnings of precision medicine, because we could take cells from everybody in the room, take them, turn them into induced pluripotent stem cells, and have an endless supply of cells to study your genetics, but also potentially in the future to provide you with cells that would allow you to be able to be treated for serious degenerative diseases. So rejection-free transplantation is a long-term goal here, although a long way off, I would say. The most important short-term goal is that these iPS cells allow us to model human disease in the petri dish and think about screening for new therapies. So, of course, as we know, Shinya Yamanaka was awarded the Nobel Prize in 2012, and I always like this because this is sort of how science goes, along with the person who first got me interested in developmental biology when I was an undergraduate in Oxford, John Gurdon, who was the first, of course, to show that in frogs you could use nuclear transfer cloning to reprogram an adult nucleus back to the beginning of development. So this is fundamental basic science, but hugely influential going forward. So these cells, these pluripotent stem cells, have rapidly opened up new vistas in regenerative medicine. And people often say, well, you know, there's a lot of hype around pluripotent stem cells. We've heard a lot about them. Are they really moving forward to the clinic? Well, the answer is, I think, yes. Just really in the last 2 or 3 years, I've become convinced that we are seeing some of these discoveries from pluripotent cells moving towards the clinic. And this is just a quick list of what's going on now around the world and the diseases and syndromes and degenerative problems that have been addressed with potential cells from pluripotent cells. Spinal cord injury, macular degeneration, Parkinson's. There's an international consortium, including people here in the UK, that's very close to a phase 1 trial using dopamine neurons. Diabetes, insulin-producing cells. And exciting but very difficult, using ES cells to generate real heart muscle to repair. And that's going on, again, in good preclinical studies. So just to say, I've been changing my slides on the run here, because actually just last week, this announcement came out that in Japan, this is the first clinical use of iPS-derived dopamine neurons to treat Parkinson's patients. So the first patient has been treated with these cells in Japan. So things are really moving forward, and we're going to see these treatments moving through clinical trials and hopefully into the clinic. But as I said, really, the biggest use of iPS cells right now is this concept of being able to study people's disease, model disease in a dish. And so we got involved in this, and this is because I'm at the hospital for sick children. So I'm going to give my research here as an example of what you can do with iPS cells to model disease and help develop therapies. So cystic fibrosis, a single gene disease, recessive disease, causes, the gene was cloned in Toronto at Sick Kids in 1989. It encodes a chloride channel, a CFTR chloride channel. And over the years, since 1989 until very recently, improvements in survival for kids with cystic fibrosis were all related to improvements in treatments of the symptoms, but not addressing the protein. Recently, there are drug treatments based on repackaging the CFTR channel and improving its function that are in the clinic. However, they don't work on all patients, even with the same mutation, and they're very expensive. So there's a big desire to really have a way of telling which patient is going to respond to which drug. So a personalized test before you give the drug to the patient. And so what we have been doing in the lab, and this is really work by a research associate, Amy Wong, is taking embryonic stem cells and then iPS cells from CF patients and driving them towards differentiating into ciliated CFTR-expressing lung epithelial cells, and then using those lung epithelial cells to test drugs. So just here, show you proof of principle. At the top, we see that we have wild-type ES cells, tight junction epithelium expressing CFTR on the cell surface, CF mutant cells, no CFTR on the surface, treat them with drug, CFTR comes back on the surface. So we can measure the response. Now we've transformed this into a multi-well plate system, so you now can take individual patients and look at their response to an agonist of the CFTR protein. And you see here's two patients, same mutation, different patients. One responds to the drug, one does not. So we're seeing now these individual responses. And of course, now what we're doing is really comparing the cell responses in culture with our colleagues' work in the clinic who are seeing the patients and their responses to drugs. So this is really moving forward through this large-scale study that brings together many groups across the hospital to really essentially try to get to a point where we get the right drug to the right patient. And again, this is kind of approach is going on worldwide with many different diseases, and even in cystic fibrosis, there's an even shorter term assay that's been developed by Hans Clava and Beck Beekman in the Netherlands, where they take primary cultures from the gut and make little gut organoids. So organoids are a new way of beginning to have new models for human disease. What we see at the top here is these little cysts, these little gut organoids, again treated with the agonist. They swell because the CFTR channel functions. The CF mutant cells don't swell. Treat them with drug and they swell. And they're actually now beginning to use this clinically to test which patients to receive which treatment. So this is really then moving towards precision medicine. So we're looking at personalized drug therapies based on modeling disease in the dish and across different diseases. We're also seeing stem cell-derived therapies. But really, this is all very well. If we know that patients have a genetic disease like cystic fibrosis, why are we still struggling to try to replace their cells with stem cell therapies or to treat the patients with drugs that they're going to have to take for the rest of their lives? If we know what the gene is, why don't we just fix the gene? So, gene therapy, gene replacement, or gene correction. Can we do that? Well, of course, you know what I'm going to say is that we have a new transformative agent here, another game changer after stem cells. The game changer is really CRISPR-Cas9 gene editing. And this is a tool that was first discovered in bacteria. It's how bacteria fight off viruses. And it was discovered by people working on yogurt, because if your yogurt bacteria dies and it gets infected by virus, you're in big trouble. And so the companies put a lot of effort into understanding how bacteria fight viruses. And they do it via this nuclease, the Cas9 nuclease, which cuts DNA, makes a double-stranded cut, a very precise positions determined by the binding of this guide RNA. And now, as people have worked through this and turned it into a tool that can go out of bacteria into many cells, you can essentially design the guide to direct that cut to wherever you want in the genome of whatever organism you want. And so it has three, actually there's a missing piece there, three very important characteristics that one has to bear in mind. It's very specific, more than most other kinds of approaches, very efficient, and, I should have said, cheap. So there are other nuclease systems out there, but the CRISPR-Cas9 is cheap, and the founders of this technology, Jennifer Doudna, Emmanuel Charpentier, Feng Zhang, George Church, and others, have really democratized it and made this available to everyone worldwide. And you can genetically modify plants, animals, bacteria, whatever. But in the context of having personalized therapies, we can start now really thinking about somatic gene therapy, not just adding genes, but potentially really manipulating the genome within enough cells in the body to have a potential cure for a disease. And this is, again, moving extremely fast forward in the clinic in many different areas. This slide just gives you an idea of just some of the different diseases for which people are thinking about ways that we might use what we would call somatic gene therapy, editing the genes within the person who has a disease. And they really cover a wide range. So cystic fibrosis is certainly one, where you might imagine getting an adenovirus into the lung and fixing, repairing the gene in enough cells to recover function. You can think about hemophilia. These are also moving to the clinic, where you would take, again, adenovirus and correct the hemophilia mutation in enough cells to get enough function. People talk about, in the long run, maybe you should go in and knock out PCSK9 and adjust your cholesterol levels. Skin diseases, epidermolysis bullosa, and these, I think, are really interesting ones, and the blood system's probably the ones that are moving fast, because there you can take out the cells, edit them outside the body and put them back. So sickle cell disease and beta thalassemia, the idea here being not actually to edit and repair the mutation, but to reactivate the fetal globin gene. So this would provide enough function to allow the patients to really have a much more normal existence. And again, these are moving rapidly to clinical trials. Many companies and many organizations around the world really pushing this forward at the somatic cell level. So, all good. But, let me just show you this. My favorite of where this is moving, recent work from Eric Olson's lab, suggesting that you may even be able to do this for Duchenne Muscular Dystrophy, which I'd always thought was a target that would be impossible, because every muscle in the boys' bodies are degenerating. So would you be able to get enough gene repair across the whole body to be able to at least delay the progression of the disease? And again, not yet in humans, but in mice and in dogs. This is recent work in dogs in a preclinical model. Olson's lab has been able to show that they can repair this mutation here that causes an out of frame. They can cut it out, reframe, put the gene back in frame, and get enough really quite amazing recovery. Not every cell, of course, but using an AAV virus, they're getting across different muscles in the body this potential repair. So we're seeing this really attacking diseases that I would have thought were impossible only a few years ago. So if we can edit out genes in patients with disease, why would we do that? So each stage, why would we do drug therapies if we can correct the gene? If we can correct the gene in the patient who has the disease, why don't we edit it out before they get the disease? So this brings up the issue of germline gene editing. So we've all seen these kind of pictures. This is the designer baby, where using CRISPR-Cas9 in the early embryo, you could envisage potentially removing the cystic fibrosis mutation, repairing it. Huntington's disease, other single gene defects could be repaired in the early embryo, and then all the cells of the baby and the next generations would have that repaired gene. So this concept then of germline gene editing has really got a lot of publicity, and as I'll show you later, I had to change my slides again. But why am I interested in this? I'm going to take this back to my research. So thinking about gene editing in embryos, the first place you go and test all these things out, of course, is in the mouse embryo. So we're back to the mouse. So can we gene edit a mouse embryo? How efficient is it, and why would we want to do that? So I want to tell you a little bit about my science now, back to the blastocyst. So I'm going to give you a quick survey of how the mouse blastocyst is put together and why we want to be able to do more gene editing to develop better tools to understand how a mouse blastocyst is put together. So we are still struggling with understanding how you get from an egg to the blastocyst and the dynamics of the process involved. We do that because we still want to understand how pluripotency is set aside in the embryo, and we hope to use that to make better stem cells either directly from embryos or by induced pluripotency. So we need better tools for gene editing. We've been able to genetically manipulate the mouse embryo for many years by slow, tedious processes where we actually do it in embryonic stem cells, make chimeras, breed the chimeras to make mouse lines, and then we can do our experiments. If we could do it so efficiently that we could do it right in the embryo, then immediately we have the mice with the mutations and the alterations we want, and the speed and the complexity of experiments we can do increases dramatically. So why do we want to do that, and what do we want to do? Let me tell you a little bit, just 2 slides really, on what we know about how the blastocyst is actually put together. So we know that there are key lineage-specific transcription factors for all 3 cell types, the trophectoderm, the epiblast, and the primitive endoderm. We know they're necessary if you knock them out. Some of them are sufficient to change cell fate. But what we have spent more time struggling with still, I would say, is to understand what's upstream, what turns on those transcription factors. We assume that there must be some signals going on as the embryo develops from a cell to a blastocyst that signals the onset of this first cell decision in the cell mass versus trophectoderm, and the next, epiblast and primitive endoderm. So what is going on? If we just look at at the expression of these proteins here, this is CDX2. It's a transcription factor that's required for the trophectoderm. In the inner cell mass here, we've got GATA6, which is a primitive endoderm factor, and NANOG, which is an epiblast factor. And they're all in a bit of a mess here. We've got double positive, single positive. So clearly, and everything we know says that this is a gradual segregation of the lineages. So what's going on? Well, we think that the first cell decision between the inner cell mass and trophectoderm summarizes everything we know, almost everything we know to date. And what we know is that actually the first decision depends on the outside cells being different from the inside cells, and the outside cells express CDX2 because this co-activator protein called YAP is in the nucleus and drives downstream transcription. In the inside cells, YAP is phosphorylated, doesn't enter, and instead of trophectoderm markers, we turn on epiblast markers like SOX2. It's phosphorylated by this serine-threonine kinase, LATS, which is in this complex on the cell membrane. And in the outside cells, because they're APQ polarized, this complex is torn apart, and therefore YAP can enter the nucleus. So we think it's a hyposignaling. We think there are mechanical signals, cell polarity. We still have to understand the dynamics of what's going on here, but at least we have an idea of what's leading up to the final lineage fate. So what about the next one? Well, I showed you already that as you go from making an inner cell mass and a trophectoderm to making the primitive endoderm and epiblast, you start with a kind of mishmash of cells, double positive, single positive, and eventually they sort themselves out. A different process from just the morphogenesis of the morula and the blastocyst. There's a cell sorting going on here. We know that a different signaling pathway is important here, and it depends on FGF signaling. And so cells that receive FGF4 turn on downstream genes like GATA6 and become primitive endoderm. Cells that don't express the receptor and don't turn on the downstream pathways become epiblast. What we don't really understand, we know the downstream responses here, we don't understand what's going on here. Is this a stochastic process? Is it a dynamic process? Are there other signals that are important here? And how do cells actually segregate themselves out of the blastocyst stage? So we still have questions. We always have questions in science, it seems. So these are just some of the things we want to understand, what we're trying to explore in the lab, my lab, and other labs as well. The roles of contractility, polarity, position, signaling. And can we actually monitor this in real time? This is a complex process. We want to study and be able to visualize the processes that are occurring in the embryo. Can you monitor transitions in transcription factor? And can we disrupt the pathways very specifically? And this is why we keep, whenever we have these questions in the lab, there's always this question of, well, we need more different mouse lines. We particularly need to have more fluorescent tagged fusion alleles and other synthetic biology alleles so we can watch the dynamic process. So this is where two postdocs in the lab, Bin Gu and Esther Posfai, took this on and said, well, CRISPR-Cas9 is great, but it doesn't work very well when injected into the mouse embryo into the zygote to do the kind of big insertions of fluorescent tags and other things that we want to see. And so they asked, well, why is that? And they, with a bit of lateral thinking, they worked out, or maybe they were just lucky, who knows, but basically, where most people had been injecting into the zygote stage, the single cell, they worked out that the best time to do this was probably at the two-cell stage, which has a longer G2 period, and homologous recombination, which is what we have to do to actually get the insertion of the fluorescent alleles into the cut made by Cas9, is known to occur largely during G2. This is also a period at which the chromatin is extremely open because this is zygotic genome activation. Anyway, whatever the reason, I have to say we don't really know if those are rationalizations, whether they're the real mechanism. The bottom line is this works and works very efficiently. So what we were making in the first place are fluorescent fusion alleles for all the different genes that we want to study in the early embryo. And this is how it's done. People, I always show this because people are always surprised. You mean you inject both cells at the two-cell stage? Yeah, it's quite easy, really. You just turn it around. we get lots and lots of emails about this whole procedure, of course, and there's always this question, is it okay if I just inject one? And it's like, well, why don't you inject both? Doubles your chance. So that's the procedure. And here were just the first experiments were done. Nothing very fancy here. For those of you who know about gene editing and the tools, we're using messenger Cas9 message, a single guide RNA, circular DNA template with sort of 1 kb arms. And just the first experiments then, two of our favorite genes, SOX2 and GATA6, going from zygote injection. GATA6 not bad, actually, but at the two-cell stage, then we really go up from very low to about 30%. And doing this approach wasn't quite good enough for Bin, so he added another twist on this to make it more efficient. In this case, what he did was to follow some work that had been done by a Chinese group in culture, where they added streptavidin to the Cas9 protein and then biotinylated the template, the homologous repair template they were going to use. So this just helps everything come together. The PCR template and the enzyme get brought in association, and this now increases your efficiency again. So what we see here is that with something like GATA6, which is our favorite gene, because it's so easy to target, we're up to close to 100% efficiency. 100% of those who are examining at the blastocysts carry the insertion allele. And in fact, we've had live mice born that are homozygous insertions. So this is now moving to extremely high efficiencies. So with this, we have been able to generate a sort of catalog. We have epiblast markers, trophectoderm, primitive endoderm, others, all of them now going through, and again, varying in efficiency between 10 and 50% and up to 100%. So now we're showing that this can be at efficiencies that start you thinking about germline editing. So Just to show you a couple of pretty pictures, what we're seeing here is an image of the embryo going from the moria to the blastocyst. The green is CDX2 tagged, and then you'll see the red, that's SOX2 coming up in the inside cells, but you'll see they never seem to overlap. So we do think that CDX2 and SOX2 really are balanced out, as shown in that model in terms of the role of the hippo signaling pathway. This is just, we're now combining more markers, so let's have 3 at once. This is now in collaboration with our friends with the light sheet microscope at Genelia. And now you're seeing the detailed analysis now. You're seeing the white, which is the CDX2, being segregated, and you're seeing within the inner cell mass, again, this complex, very dynamic process that eventually leads to a segregation of the endoderm and epiblast. But once you start looking at this, you see it's not that simple. So more analysis ahead, certainly for this. So there's lots of things we can do in the mouse with this, and we and others are obviously going to do this. It just moves us forward in mouse genetics. New revolution in mouse genetics, all that ESL stuff that we did for many years, those cells are sitting in freezers. Everybody is doing it with gene editing. We can use synthetic biology tools. We think we can do genetic screens right in the embryo by efficiently making stop codons in the genes. We can have multiple reporters, not just in the embryo, but in other tissues as well. Maybe the people who are looking at the cell atlas might want to tag every gene, so we could look at that in different ways. And of course, then the question becomes, can you extend it to non-human primates, or indeed to humans? So I am not saying that the approaches we're doing in the mouse, the 2-cell injection, is necessarily going to be something you would want to move to the human, because even at the most efficient approach that we have, we are not at 100% efficiency. If you want to genetically correct a mutation in a human embryo, you need 100% efficiency every time, and you need to be sure that it's safe and you have no off-target effects. So, but it does mean that it's so different from where we were just a few years ago that the concept of doing human germline editing is a realistic possibility. Everything is improving. Every day, new approaches are coming out to improve the efficiency of this kind of gene editing. So it would allow individuals to have genetically related children without passing on a known risk of a genetic disease. Has potential, if we wanted to alter the predisposition to diseases like cancer, heart disease, Alzheimer's. And, of course, potential for human enhancement. Back to that picture of the designer baby, one of them was, improved athletic ability. I think we can all think of a few genes that one could edit, that one would know would actually do that. So enhancement is not out of the question in terms of the possibilities. And then these, of course, raise all the sort of ethical concerns that we as scientists and society have to start discussing. So where do you draw the line? Well, I was part of the first International Summit on Human Gene Editing in 2015. It's me on the stage, which came out with this statement that basically said it would be irresponsible to proceed with clinical use of germline editing until we know it's safe and efficient. We need to know the risks and potential benefits. And there's a broad societal consensus, whatever that means, about the appropriateness and regulatory oversight. At present, these criteria have not been met. After that meeting, the National Academies of Science was one of the many working groups that around the world have considered this. The NAS spent a year with many, many stakeholders. I was part of that process as well, and came out with a very influential document that I have summarized, pages and pages, into a sort of traffic light system. The conclusion was that gene editing for basic research, because it is a hugely important basic research tool, as I've just shown you in the mouse embryo, should be done in human cells and in human embryos. And here in this institute, Kathy Nyakin is using gene editing to study the same kind of questions in the human embryo that we're addressing in the mouse embryo. That somatic gene editing to treat disease has huge potential, and that would have no particular different regulatory concerns than current gene therapy protocols, might even be safer. Germline editing, I put amber here, which means not a complete ban, but think very carefully. Germline editing to treat serious genetic disease when no alternative exists and all the safety issues have been addressed. So not a no, but very high bar to pass before that should go forward. And then a red to either somatic or germline editing for enhancement purposes. Now, define serious genetic disease, define enhancement. These are the issues. So that was the call from that group. And I think over the last year or so since that's come out, if anything, worldwide, including here in the UK, there has been various surveys of the public and so on to say that people are beginning to be a little more understanding that germline editing might be considered one day. So the immediate sort of gut response, oh no, we shouldn't do that, has softened. But nobody is really proposing that we're anywhere near safety and efficacy to move this to the clinic at this time. So you know what I'm going to say now, is that as of yesterday, so that's what I got in my talk, and then I had to add a slide. So just yesterday we heard that Jianshu He in China has claimed to have produced twins in which he used gene editing, just the way I showed you in the mouse, to make mutations in the CCR5 gene, which is the receptor for the HIV virus, which in theory should prevent those babies being infected with HIV. It's pretty clear that this contravenes everything I just said about germline editing as in the NAS report. This is not a serious genetic disease. It is an enhancement. This is definitely an enhancement, and in fact, potentially dangerous enhancement, because loss of that protein also makes you more susceptible to other viral infections, so we don't know the consequences, even if it's true. And it's not correcting a serious genetic disease. And it's unclear exactly what ethical permissions he had. His university has certainly disowned him and is calling for an international investigation. And the hospitals in which he claims to have had ethical review don't seem to agree, and the experiments were done in other hospitals. And this is really difficult, and it's particularly difficult for our Chinese friends. Because you saw in that first genome editing summit that actually on the stage was Xuan Xingpai, who is one of the Chinese leaders in the Chinese Academy. So everybody has been working on an international consensus here. And so the Chinese scientists feel that this is really obviously something that they want to object to. So there is an announcement signed by Chinese scientists which basically says this is unethical, it contravenes everything that we've been working for, and that China needs to be sure that they can set in regulations to prevent this happening in the future. But there it is. And so it occurs at exactly the same time. So I'm here and not at this meeting, so perhaps just as well. And that's the second International Summit on Human Gene Editing is taking place as we speak in Hong Kong. And that doesn't seem to me to be a coincidence that this report was put out at the same time as this meeting. So the question is, will we get a new consensus emerging from this meeting? Can we move forward? Because this is going right against everything that anyone around the world is working for. And it happened in China. It can happen anywhere. It's very hard to completely control what is, as I said, an efficient, easy, and cheap approach to modifying the genome. So that's it. We've gone from the blastocyst to stem cells to editing to ethics, but it all begins and ends with the blastocyst. And I just have to thank for some of the research my own research that I talked about, Amy Wong for the lung work, Esther Postfire and Bin Gu for the 2-cell editing, our collaborators at Genelia for the imaging, and our collaborators at Karolinska for some of the RNA-seq on single-cell analysis that is part of what we do to understand how the mouse embryo develops. So thank you.